Hi, good afternoon. I'd like to thank the organizers of the conference for inviting me to come and talk to you today. So biomedical studies that are published in the primary literature really serve as the basis for development of therapeutic drugs to treat disease. So maybe more than any other area of science, whether or not these studies, these initial findings are solid and reproducible, has a measurable cost. In 2015, Friedman and his colleagues tried to estimate what that cost was. So given that $56 billion are spent annually in research and development for preclinical studies, and using a pretty generous estimate of 50% reproducibility, he estimated that about $28 billion is spent every year on studies that are not reproducible. And so maybe it's not surprising then that it's been the biopharmaceutical scientists that were the first to sound the alarm about the reproducibility crisis, in that they were trying to reproduce studies internally, and they were finding that most of them were not reproducible. And this ranged from one ALS Institute study of 0% that retested 70 drugs from 221 independent studies, to 11% by Amgen scientists Begley and Ellis that attempted to replicate 53 landmark oncology studies, to only as high as 21% in a study by Bayer scientists that did in-house target validations. So for all of these studies, what I'm talking about is results replications. And Goodman and his colleagues defined results replications as obtaining the same results from conducting an independent study that uses as closely as possible the protocols from the original. And they also said that reproducibility is the minimum necessary standard condition for a finding to be believable and informative. And one of the most common forms of feedback that we get for these kinds of replication studies is that science is self-correcting and that they're completely unnecessary. But there's really little evidence to support that idea. In this survey, they found that 50% of scientists report that they had experienced an incident of irreproducibility from their own lab, but only 30% of those respondents said that they had published that result. So why don't scientists publish their failures and enable self-correction? Well, that's starting to change. We don't have a culture of replication. Replication experiments are still not the norm. They can create issues of retaliation of community of peers. So these are the same peers that sit on tenure committees and make tenure decisions. They also sit on panels that make funding decisions. And it can be really difficult to interpret replication studies, especially if they weren't designed initially as a formal replication. So I work for a company called Science Exchange. We're an online marketplace that allows researchers to find labs that that provide experimental services. And our CEO, Elizabeth Irons, has really been on the forefront of designing replication studies, both for biomedical studies like the Reproducibility Project Cancer Biology, as well as the Prostate Cancer Foundation, to direct statistical replications of HIV studies that's funded by the 3IE and Gates Foundation, as well as private pharmaceutical replications where pharmaceutical companies come to us so that they can confirm a result before they invest more money in further research and development. So one of the projects that's gotten a lot of attention is the Reproducibility Project Cancer Biology, and this is a collaboration with Science Exchange and the Center for Open Science, and it's funded by the Laura and John Arnold Foundation. So this project identified 50 high-profile preclinical cancer biology studies to perform direct replications. And so some of the key features of this project that could be best practices for any replication study are these. So all of the original authors were contacted at the beginning of the project so that they could give input about the protocols that we were planning to use, and we also asked them for any unique non-commercial reagents for the project. Of all of the authors that we contacted, 62% of those provided either information or data. So for 19 of those studies, we got data from the original authors. And the rest, the 38%, were unresponsive, and then a few opted out. So once we had written the protocols and our analysis plan, these registered reports in collaboration with, so all of these studies are published in eLife. So they were peer reviewed. So before we had started any experimental work, and we've heard a lot at this conference about pre-registration. So the registered reports were the pre-registration that had all the protocols and the analysis plan, and often the original authors were contacted to be reviewers on those registered reports. So they had another opportunity to make comments on the plan. And as part of that, we would do power calculations to make sure that the sample sizes for the replications were sufficient to detect an effect if it existed. And then one of the reasons that Science Exchange was an obvious partner for this project was that we were able to leverage the network of labs on Science Exchange to do the experimental work. So these are independent labs, and we chose labs that had specific technical expertise for each of the projects. And then once the experimental work was done, the replication studies were again peer-reviewed just to make sure that we had stayed with the protocols that we had originally proposed. And the important part of this project is that all of the protocols, the results, and the data are available, they're open and available to everyone. And they're maintained on the Open Science framework that's part of the Center for Open Science. And then as part of the register report process, then all of the findings would be published regardless of the outcome of the studies. So this project is still ongoing. Of the 50 original studies that were identified, 31 have been written as registered reports. 29 were accepted and published, and 2 were rejected at the registered report stage. We've completed now 8 of the replication studies. 5 of those have been published. 2 others have been accepted but not yet published, and another is still in peer review. And then the rest of the 21 studies are ongoing in the labs and are still in the process of finishing experimental work. And then we have 19 potential studies that are on hold. So at the end of all of these studies, once all of these replications are finished, we plan to do a meta-analysis, which is really the interesting part of this project. So all of the biopharmaceutical studies that I showed you at the beginning, they reported reproducibility rates, but they didn't make any of those data open to the public. So we didn't really know which studies they had replicated. And in this project, everything is open, including the data for reanalysis. So once we do the meta-analysis, we can ask questions like, what were the main roadblocks for conducting the replications at all, which is a really interesting story. And how much did they cost on average, and how long did they take? And as well, what are the factors, if we can identify any of those that are associated with studies that were reproducible versus those that weren't? So I don't think very many scientists would argue that the system is completely broken, although I think I may have heard that while at this conference. We make scientific discoveries all the time. We develop drugs that treat disease and work. But I would argue that improving reproducibility rates makes us more efficient than a self-correcting model. For biomedical research in particular, improving the speed with which solid discoveries can be moved forward in development is particularly important. So how can we encourage a culture of replication? I'm going to make 3 suggestions. So the first one is to reduce the reliance on contacting the original authors. For all of the studies that we've done so far, we it was required that we got information from the original authors. In none of the cases was there enough information in the method section for us to be able to really do a true replication without the original author's input. So we can already do this. So there are methods for us to be able to store information like protocols and data, and some journals now are allowing that as well. One of the other really important things is uniquely identifying reagents in method sections. So the resource identification portal allows you to uniquely identify all of the reagents that you use in a method section, and that's really important, especially for these preclinical kinds of studies where specific antibodies are very important in doing the replication study. And then lastly, there are already repositories for scientific material, and those include things, places like AdGene, JAX, and ATCC for cell lines. So the more the authors can make those reagents available will lessen the requirement that they be contacted for follow-on studies of their work. The second suggestion is dividing research studies into different categories and identifying studies in each category. So the first is what we're used to, exploratory novel studies. Those are the ones that most of the literature is filled with. So those are multi-year investigator-initiated grants. And I would suggest a second type of study, which are confirmatory studies. Those would be conducted by technical experts. So they might require different types of labs and scientists, for example, using core facilities to those kinds of confirmatory studies. And they would require rigorous QC procedures that would be identified ahead of time. And then lastly, the most important factor that can promote replication studies is funding. There's almost no grants that fund replications. It's encouraging to see that the Netherlands is kind of on the forefront of this idea. They just this last year allocated $3 million specifically for replication studies. So thanks for your attention. I'd be happy to answer questions.
Go. So the floor is open. People are lining up.
Hi, André Knot-Neris, the Netherlands. Thank you very much. Very clear and informative presentation. Compliments. My question would be, if you do an excellent original study, it may be an observational study or a pharmaceutical trial, and it gives a clear answer, a positive answer, or when the power is sufficient, also a credible negative answer. Is it then ethically justified to replicate exactly that study, burdening patients, while there is no equipoise anymore, while the hypothesis has already been confirmed? I don't know the answer, but I think it's interesting to comment on those ethical applications, especially when we talk about research integrity. So could there be a tension between replication and being justified to burden new patients with research risks?
So if I understand your question correctly, you're asking about like first-in-human trials, so those kinds of replications? Or just any?
I think it's a general question. Also in observational research, you use resources. You may burden patients or other research subjects. But let's take a trial. If you do a very good trial, which is conclusive in the positive or the negative sense, could there be an ethical conflict with just replicating exactly that same study? Would it survive the institutional review board?
I see. So these were really focused on sort of preclinical studies. So I guess the same question has been asked for preclinical studies with respect to using animals for replications, for example. And I guess my answer to that would be that I think that the fact that so many studies in the literature that have gone through peer review and have gone through, IACUC approval are not well supported with replication data suggests that might be less common than we would like to think. I guess it's sort of identifying those factors of the study that you're describing. If we can have more confidence that studies are designed in that way, then I think that's what we learn from with these replication studies. And maybe we get to a point from doing the replication studies and improving the way that we do science to get to the point that you're talking about, where you wouldn't necessarily always need a confirmatory study to believe the results.
Hi, I'm Jean-Philippe de Jong. I'm from the Netherlands Academy of Sciences. You mentioned briefly that it would be interesting after the cancer biology replication effort to do an analysis of the costs of that effort. Would it be feasible to do sort of a kind of cost-benefit analysis, and what would be on the cost side? And this gentleman already mentioned the cost side would also include additional use of patients or animals. And what would be on the benefit side? Could you elaborate on that?
Sure. So when I mentioned cost, I was talking about like the actual cost to do the replications. I think there's a general sense that this would be way too expensive to go into. So I think it would be good to get some real data about how much the replications actually cost. I think you're talking more about, in general, estimating other kinds of costs. And yeah, I'm not sure from this meta-analysis if we could estimate the benefit necessarily. We can only say that we have a better sense of what the reproducibility rate is in this set of studies, and then we can hopefully try to use that information to improve the way that we do science. But I don't know that you would be able to make a direct benefit from that analysis of these studies.
Yeah, maybe the evidence base in your sample would be too small, but I think that will be the really interesting thing to think about it in terms of cost and benefits of doing replication studies and seeing what the direct benefits of a study will be on the evidence base or towards the drug pipeline or patients.
Yeah.
But also the broader impact of doing replication studies, changing the culture of replications, etc.
Yeah, and I think the replication program that the Netherlands is starting that would be a mechanism by which you could see whether you could actually change, you could move the scientific community in one direction or another. I think it needs to be sort of baked into the way that we write grants and get funding.
Okay, thank you.
Okay, time for a final question. Short one and a short answer, please.
Yeah, the question I think is quite short. I'm Joanna Johnson from the University of Miami. Nicole, you mentioned about the meta-analysis that you were thinking about doing or going to go on to do. Do you have any sense yet about what some of those causes of irreproducibility are?
Well, from the first 5 studies, which is very small sample size, I think it's going to be that meta-analysis is going to be really interesting because we've already seen lots of examples of different reasons why replication studies can be difficult to interpret. So, for example, the model didn't replicate, the cell line didn't behave in the way that we expected in one study. I can say that is true. So I think that we'll definitely be able to identify some key components that need to be addressed before doing a replication, which I think will be really important. I can't really draw conclusions about what makes a study reproducible or not.
Thank you very much, Nicole.
Thank you.